There you go. And because I am recording, I just wanted to let you know, I mean, most of you look like you've got your video off, which is fine. And so please feel free to keep it off, because this will be recorded. If you have concerns about that, just leave your video off. Diego is going to talk for, I don't know, 20, 25 minutes, something like that, maybe half an hour. And then we'll open it up for questions. So with that, it's my great pleasure to pass it over to Diego. I'll go on mute and you can take control of this screen.
Well, thank you so much. Let me share the screen now with everybody. Is this okay for you? Like, can you see it?
I can see it, yes.
All right, fantastic. Let me see. All right, so for those that are in the audience and didn't see the paper, it's called The Indo Pacific Origins of Silky Shark Fins in Major Shark Fin Markets, Highlights, Supply Chains, and Management Bodies Key for Conservation. So I'm going to walk you through what we did for this particular paper. There was a paper very recently published, like a month or two ago, that has shown that since the 1970s, the relative fishing pressure on oceanic sharks has increased 18-fold. So that's a very, very sharp decrease in the last decades. And unfortunately, that has an impact in the global populations of oceanic sharks with a decline of 71% since the 1970s. And that obviously, with that fishing pressure and that decline, that basically means that there's a higher extinction risk for these species. Here you can see that back in 1980s, there was a few species in the threatened categories of the IUCN. And now in 2018, in the last assessment, we have many, many more species in these threatened categories, especially the endangered and critically endangered categories. So most of the fisheries that interact with these oceanic species are under the purview of RFMOs, or regional fisheries management organizations. We have ICAT in the Atlantic, IOTC in the Indian Ocean, WCPFC in the Western and Central Pacific, and IATTC in the Eastern Pacific. And then these management bodies, or these RFMOs, have some of them have particular regulations or management for some of these highly mobile and oceanic shark species. Also, CITES, the Convention on International Trade in Endangered Species, has recently listed a lot of these oceanic species and globally distributed species on Appendix 2. And here you can see that the mostly widely distributed species and the most common in trade are in CITES Appendix 2. So here you can see the makos, you can see the oceanic whitetip and the hammerheads and the silky shark. So all this group is mostly on CITES already. Well, here mostly the coastal species that are still common in trade haven't been listed on CITES yet. So the reason for this is also that the oceanic sharks are some of the most common shark species found in the international shark fin trade. This particular data is for mainland China, but we've also found that the CITES listing of these species since 2003, the enforcement and the compliance of the CITES regulation hasn't been as we would expect after the enforcement started. So here in this bar plot, you can see the legal importations into Hong Kong in 2015, which is the first year of enforcement after the hammerheads and the 2013 CITES listed species got listed, which was late 2013, and the enforcement started late 2014. So this amount of imported shark fins is just 0.4% of the total amount that was imported that year into Hong Kong. So you would expect CITES listed species to be rare in trade. And then what we found, and you can see here in the bar plot on the right side of the slide, is that the relative proportion of hammerheads in 2014, which is before the enforcement, 2015 and 16, did not change over time. And then this sampling is still occurring. Well, I'm going to explain a little further in a few minutes. And this proportion is not changing over time. So there's definitely high volumes of fins of cited listed species still coming to Hong Kong, which is the largest shark fin importer in the world. And here you can see like the different routes that these fins take to get to Hong Kong. Hong Kong exchanges fins or trades fins with around 83 nations every year. So it's quite an important site for the conservation of these species. And as I said before, we suspect that there's still a lot of illegally traded shark fins coming into Hong Kong every year. So what we did since 2014 to now, basically, we're sampling the shark fin markets of Hong Kong. But the problem is that if you buy the fins as they are in this right hand picture, for example, they're quite expensive. A kilogram of a particular species of ray actually can be a little over $1,000 US. So if we would sample the large fins, there's no budget that we could have to make a significant survey. So we use shark fin trimmings, which are just once the fin arrives to Hong Kong, it arrives as a form of unprocessed and dry, and then they process it. And at the end of the processing process, they cut the fin in different ways just to make it look better, and like they remove the cartilage at the base and just trim it here and there, and then they sell those trimmings for much cheaper. So we buy these fins, these shark fin shark fin trimmings from the markets, and that gives us a proxy of what is being sold in the from different vendors. So in Hong Kong, we started in 2014, and the published data is until December in 2017. What we do is we select 10 random vendors every month, and then we go and purchase 2 bags of these shark fin trimmings per vendor. The first year we did that twice a month, and then the second and third year we did it just once a month, and we collected and analyzed over 9,200 samples. Then we went to Guangzhou, which is the largest shark fin trade hub in mainland China, and we did the same sampling from June 2015 to August 2017. Same thing, 10 random vendors, 2 bags per vendor. We went there every 2 or 3 months, and we collected 2,000 samples, and we analyzed them to the species level to see what the species composition was in these different markets. And here you can see kind of like the top species in the trade. And one thing that is like to highlight is that the silky shark, which is vulnerable on CITES and is an oceanic species, is the second most common species in trade. So this species has been consistently the second most common for decades, although this sampling that I just told you about started in 2014. There's records of similar samplings conducted at the early 2000s, and silky shark was the second most common species. So it's been in that position for over 2 decades. Is the most common species taken in pelagic tuna fisheries in tropical and subtropical regions. As I said before, is vulnerable globally by the IUCN, and it was listed in Appendix 2 by CITES in 2016. So these 4 RFMOs are kind of like the managing bodies that have the fisheries that interact with these species the most. And ICAT in the Atlantic in 2011 banned the retention and the landing of silky sharks, which was the first RFMO to do like a managing or like a regulation for these species. Then WCPFC did the same in 2013. And in 2016, IATTC in the Eastern Pacific banned the landing and the catch and retention of silky sharks in the purse seine fisheries, although the long lining fisheries in this region of the world can still catch silky sharks. And IOTC does not have any regulations in place for these species for now. So we went back to 2015 and found a paper by Clark et al. That did the global population genetic structure of the silky shark. And the good thing about this paper is that it showed that globally, based on the mitochondrial control region, there are two different clades, one in the Indo Pacific and one in the Atlantic. So this basically means that we could genetically identify and isolate the sharks that were coming from the Atlantic. And we use that baseline of the genetic population structure of these species to assess the relative contribution of Atlantic silky sharks under the management purview of ICAT and Indo-Pacific silkies to the two largest shark fin trade hubs in the world, which will be Hong Kong and Guangzhou, as I already said. So for this paper, we took the samples collected by Clark et al. In 2015, which are the black squares here in the screen. But we increased a little bit the sampling effort and the geographical coverage of the sampling just to fill out the holes that the other paper had. So for example, we collected more samples in the Eastern Atlantic, the Indian Ocean, the North Central Pacific, South Central Pacific, Papua New Guinea, Taiwan. And then we took from our sampling from Hong Kong, from the species that we identified already as silky shark, we took 604 samples that were collected throughout the year. So some from 2014, some from 2015, 16, and 17. So all these samples were collected throughout 4 different years and different vendors as well. So because these samples and these shark fin trimmings in Hong Kong are highly degraded because they have been processed, which is they use a lot of chemicals and bleach. So that degrades the DNA a lot. So for the mitochondrial region, what we did, which is around 1,100 base pairs, we designed internal primers to have different size amplicons in the most informative region of the mitochondrial control region to be able to assemble the haplotypes based on these 3 different amplicons. What we found out is we yielded a total amount of 96 haplotypes, but not all 604 samples worked. Here you can see that for Hong Kong, we had 169, and for mainland China, we had 164. That is around 2/3 of our samples, because the other third, either just they didn't amplify at all, or we just were able to amplify 1 or 2 of those amplicons, not the 3 of them. So for these 3, we used samples that we had all 3 amplicons, the samples that were collected by Clark et al. And the samples that we collected ourselves. And here you can see that all the samples from Hong Kong and China fell in haplotypes from the Indo-Pacific clade and none from the Atlantic clade. Although you can also see that in the Indo-Pacific clade, we have haplotypes that are shared between the Atlantic and the Indo-Pacific population. So we were not sure if those animals were either caught in the Atlantic or the Indo-Pacific. So that's why we did a mixed stock analysis, which basically takes the haplotype frequencies that were described by the Global Population Structure Study. Here, H1 is group of haplotypes that have only been described in the Indo-Pacific, and the H7 is a group of haplotypes that have only been described in the Atlantic clade. The rest of these haplotypes are the shared ones. So here, just by looking at the haplotype frequencies, you can see that the samples that we saw in Hong Kong and mainland China, it's much more similar to the ones from the Indo-Pacific. And once we conducted this MSA or mixed stock analysis, we saw that the Indo-Pacific contribution to these markets was 99.8%. So it was quite an interesting finding, because we were definitely not expecting the Atlantic to be that low or close to 0. We also, just to add more evidence to what we were seeing, we took the samples that we were able to amplify that first amplicon, which is highly informative. And we were able to use 455 to reconstruct this haplotype network. And here we saw basically the same pattern. All the haplotypes with market-derived samples in them fell within the Indo-Pacific clade. So that gave us further confirmation that we were not seeing Atlantic silky sharks in these markets. So as just as a little abstract of what we found is just near zero occurrence of Atlantic silky sharks in the Hong Kong and mainland China markets. The Indo Pacific RFMOs, which just to remind you were IATTC, WCPFC, and IOTC, are basically the primary management bodies overseeing the supply of silky sharks to these trade hubs. And like all the fisheries management and CITES implementation should focus to this like large region. And this was in basically parallel with what you see if you check the landings reported by these RFMOs of silky sharks. So the Indo-Pacific RFMOs basically report 93% of the global landings of the silky sharks. That 7% is actually from the Atlantic, but ICAT allows the developing nations within the Atlantic to still catch some silky sharks as a food source, but there's an exportation ban on the fins and meat. So some of these countries are still allowed to catch silky sharks, but they cannot internationally export any of these parts of these silky sharks. So basically what we found is that there's high compliance with the ban on international exportation of the fins and high compliance with the retention and no landing of silky sharks imposed by ICAT in the Atlantic in 2011. We did an extra analysis, statistical analysis to see what was kind of like the threshold of the proportion of Atlantic silky sharks that we needed to pick them up in our samples, and it was a couple percent. So definitely our sampling, if they were there, the Atlantic silky sharks were within, let's say, our samples collected from the market, we should have picked them up. So that gave us extra confidence on our results that there were no Atlantic silky sharks being picked up in the markets. Let me see what's going on. All right. So the next question that this paper raised is like, what is the level of compliance in WCPFC, which has a similar no retention and no landing ban within their jurisdiction? And also in the purseine fisheries for IATTC. So if we assume that the compliance in WCPAC and the purseine fisheries in IATTC is high or as high as the ICAT one, then the supply of silky sharks is just a matter of small scale domestic fisheries that are outside the RFMO jurisdictions or these RFMO jurisdictions, or IOTC fisheries, which don't have any regulations in place for the silky shark, or just also by longline fisheries under IATTC, which are allowed to catch silky sharks so far. So another thing that we need is that kind of have more detail in the genetic population structure of the silky shark. Here we use the mitochondrial control region, but there's genomic studies that show that if we go into the genomic side of things, we'll delineate better this population structure, and maybe we can in the future pick up individual RFMOs. So not just Indo Pacific, which covers 3 different RFMOs, just like individual ones, which will help us identify better these key management bodies for silky sharks. We also need, obviously, better management and enforcement of laws that are around the silky shark. So one of those is increased enforcement efficiency, and there are tools that allows us to do that. Visual identification guides, quick tools to identify, quick genetic tools that allows us to identify illegal trade in CITES listed species, including the silky shark, which will help us to detect and deter this illegal trade. And also we need like a better monitoring of these shark landings of especially the silky sharks where they're banned in the Indo-Pacific, just to have a little grasp whether there's high compliance or not in these regions. And also like a call to IOTC to start management for these species. It's likely that a lot of the silky sharks that we're seeing in the markets do come from the Indian Ocean, since there's no management or regulation put in place. So that should be something to look into in the near future. We have done similar studies for other different species, and particularly CITES listed species. So here in the left, you can see the main results from Fields et al. In 2020. which saw that the Eastern Pacific was the largest contributor of scallop hammerhead shark fins to Hong Kong, which around 63% of the analyzed sample. So the Eastern Pacific is definitely a region of concern and a region to highlight and better enforce all these regulations. And I recently did the pelagic thresher shark as well with samples collected in Hong Kong and mainland China. And I found out that 85% of those samples were coming as well from the Eastern Pacific. So both the scallop hammerhead and the pelagic thresher agree that the Eastern Pacific is a region of concern and a region to put more conservation efforts for these highly migratory and oceanic species. The next steps will be to do black tip samples. So as I said before, we have all these library of samples from Hong Kong and mainland China. So the next steps will be to trace back black tip samples to a region of origin, as well as short fin mako, which will help us to have a baseline of North Atlantic makos now that they are, now that the European Union has a zero quota for them, just recently imposed. And we're also going to start tracing back scallop hammerhead and smooth hammerhead juvenile ones to a region of origin to kind of like pinpoint different nurseries that might be supplying a lot of the volume of these species into the shark fin trade as well. So what these studies are showing us is that if you combine large-scale surveys in markets and trade hubs, and you combine that with landings data and monitoring of the landings, you can combine those two data sets and have a very good idea of the dynamics of the trade in different species. You can start pinpointing areas around the world where these different species are coming from and increase conservation efforts in these different regions. So it's just an example of what you can achieve if you combine these two different samples and sampling methodologies and data sets. So for that, I think I'm done. I don't know how long did that take, around 25 minutes. But I don't want to finish before acknowledging and thanking all my co-authors for this paper, the Society of Conservation Biology for hosting this and allowing us to show our results, conservation letters, obviously, for publishing our paper, and the different institutions and that have like supported us and both academically and financially. So with that, I'll take questions if you have. That would be nice if you have any questions for this research effort. Thank you.
Excellent. Hey, thanks, Diego. That was a terrific presentation, really clear, really important results. Hey, so everyone, please, if you have a question, just unmute yourself. You just need to go to the button on the right-hand side of your name and unmute yourself. Everyone was muted automatically when you joined. So feel free to unmute yourself and ask any questions you have.
I have a question. So I'm curious why sharks like the silky shark and hammerheads, from what I see from your finding, are targeted more than the other species of sharks.
Yeah, thank you. I don't know, I can see your name, but thank you for your question. Well, the reason, at least for the silky sharks, is that their habitat range overlaps a lot with large tuna fisheries. So they're caught in like purse seine and longline fisheries as bycatch, and they're a desirable bycatch because you can sell the fins and you can sell the meat. So that's why I don't think tuna fish are particularly targeting sharks. They're targeting tuna, but the sharks are really close to tuna. So basically, that's why. And I think the same goes for all these different species. A lot of, for example, the blue shark is the #1 species in trade. Although they're targeted fisheries for the blue shark, they're also a large bycatch in marlin and swordfish fisheries. So that's kind of like a big problem for sharks because they're caught as bycatch in many fisheries around the world. And that's particularly true for these oceanic species.
Diego, just building on that, is that, I mean, we've certainly seen in our work on fish aggregation devices that the silky sharks are one of the species that does tend to aggregate high catches when you have purse seines setting on fads. Are there other species that kind of aggregate in that fashion?
I don't know. I feel like there are more. Definitely, some other oceanic sharks would do it, but it's known that silky sharks just follow those fads like crazy, and that's why they get caught in such large numbers, especially in the Pacific, where they use a lot of these fishing aggregation devices. Yeah. Any more questions?
I have another one. Sorry, you probably hear my kids going to school in the background. Were you able to engage directly with any of the RFMOs? And I'm curious, particularly how that kind of research level engagement has happened with RFMOs, because it's not an easy thing often to pull off.
No, I didn't. I haven't had any like direct conversations with them, but I feel like as 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 we progress with these kind of studies and we start looking at different species and we build up a real scenario for some of these highly priority species for conservation around the world, I think this contact with RFMOs and this engaging with them is going to happen at some point. I would be surprised if it doesn't. Once we finish all the MACOs and all these different species, I think a conversation is going to have to happen with them because I think this type of data is quite important for them, because it highlights either what they're doing good or where and what should they be improving. So it's kind of like a missing piece that we're being able to start like closing those gaps and like identifying the start point, the starting points of these supply chains was something that we didn't know before. So now we know the starting points of the supply chain. Obviously, the end point of the supply chain is definitely Southeast Asia, Hong Kong, and all these trade hubs. But there's also the midpoints of the supply chains that we should kind of like put our focus in it. But now we have both ends identified, and then that's a huge step forward for the management of these species, definitely.
Diego, how far away do you think you are from the exciting possibility of being able to define stocks of these down to the scale of an RFMO? I mean, that would be quite a significant
step that forward. Would be exactly. Yeah. There was a paper recently by Kraft et al. And then they did some genomics work in the Atlantic. And within the Atlantic, you can also separate some of the South and North Atlantic. So I suspect that if I do the same for the Indo Pacific and Indian Ocean, some of that like big region that we have is gonna start being separated. So the question would be how difficult or how easy would be to do genomics work on these highly degraded samples. They're extremely degraded. So just to identify them to a species level, we designed back in 2017 like a mini barcoding protocol. So I have the feeling that it could work. I just have to try with these highly processed samples. We're definitely trying with some bull sharks as well, some of these genomic work. So depending on how that goes, we can take the route for silky sharks. And yeah, if we can separate those 3 RFMOs and give a specific contribution, that would be huge. So I don't know how far, but hopefully soon.
The questions.
So yeah, I have a question. This study is based up until 2017. But what do you think about the current situation with COVID? Do you think it affects the shark finning in some kind of way?
Hi, yeah, Mariel, thank you for your question. So the published data is up to 2017, but thankfully we've been able to keep our sampling every year, so it's ongoing at the moment. And I I honestly couldn't like put my finger on an answer for your question, but my guess is that it hasn't affected sharks neither a negative or a positive way. I think like the fisheries are still going. So I wouldn't expect like the fishing pressure to decrease just because of COVID, because the fishing fleets are still out there. So I don't think it has any impact, either positive or negative, on sharks, but I don't have any evidence to support that. It's just my guess.
Anything else, guys? If not, well, I'm sure Diego would be happy to take questions directly over email if you have any follow-up thoughts or questions. And
Yeah, for sure.
Thank you very much, Diego, for presenting here and for publishing in our journal. Like, this is exactly the sort of work that I love to see in conservation letters. And thank you everyone for attending this SCB Emerging Issues seminar. I think we're going to try and do one a month or something like that. So
thank you for hosting it and inviting me. That was awesome.
No, pleasure. And I would say, on behalf of SCB, if anyone in the audience, if you have any ideas for things that you'd like to see the journals or the society doing around publications, please feel free to reach directly out to any of the editors in chief. We'd appreciate it. And have a great day, folks.
All right, you too. Have a good one. Bye, everybody. Thanks for joining us.